Thinking about the major advances in Parkinson research over the past 5 to 10 years or so, I would say that the driving force has come from studies in genetics of Parkinson's disease. Ordinarily, Parkinson's disease is not an inherited disease. Generally, it occurs on a sporadic basis. It does not run in families. Rarely, though, it can. And when it does, it creates an opportunity to determine the specific genetic mutation that has caused the disease in that family and similar families. And what that does for you is it gives you finally a handle on the disease.
In the last decade and a half, we've discovered for the first time genetic forms. We didn't have those before. There aren't very many of them. The most common one is called LRRK2, and it's only about 1.5% of the population.
LRRK2 has very well-defined properties as a protein. So it's provided a number of very important leads in understanding Parkinson's disease. And I would say it is one of the major research frontiers today in trying to understand the cause of this disease.
This is a complex disease, and it has both a genetic and environmental component. We're now discovering more and more genetic risk factors, things we inherit, but they don't necessarily do anything by themselves. Genetics loads the gun, environment pulls the trigger.
The very first genetic cause of Parkinson's disease to be discovered, and this was back in 1997, was in a gene called synuclein. It was found that not only do mutations in synuclein cause this disease, but if you've got too much synuclein, you get Parkinson's disease. Every gene that we have is regulated for how often it's turned on and how much it's turned on. And it's regulated by a part of the gene called a promoter. In individuals who have the promoters that put you at risk for Parkinson's disease, mild head injury, can put individuals at risk. It doesn't necessarily cause the disease, I want to be clear on that, but head injury does increase the risk of developing the disease, and that in combination with these promoters that lead to a greater activity of synuclein yet again adds to your risk.
One of the major breakthroughs in the last 4 years has been the ability to take adult human cells and change those cells into stem cells. Then you can take these stem cells, these newly born stem cells, and take them forward again, develop them into dopamine neurons. Dopamine neurons are the type of neurons that die in Parkinson's disease. So for the first time in the history of medicine, we'll have these patients' dopamine neurons in a dish. We call it Parkinson's in a dish. We'll be doing clinical trials in a dish. It's revolutionary.
In recent years, we have developed an important focus in our research on Parkinson's disease in studying what are called the axons in the brain. These are almost like the cables, the fiber optic cables, let's say, between one brain cell and another. And this is what the brain depends on for its intricate connectivity. We have found that contrary to what has been believed in neuroscience for decades, this is one of the longest held beliefs in neuroscience, has been that in the mature adult brain, once axons are lost, they can never be regained. And this, of course, has been a stumbling block to treating patients with spinal cord injury, patients with stroke. It's been felt to be a complete impasse. Well, in our efforts to try to protect axons, we found that similar approaches are able to actually induce the regrowth of axons in the mature brain. What we do is we'll use a neurotoxin to damage the neurons that are ordinarily damaged in the disease. We put in a toxin that destroys the axons and kills most of the cell bodies as well. We then wait for a period of time, several weeks, months, and then we have developed a gene therapy-based approach where we use what's called a viral vector, which enables us to put a new gene into the brain cells that have survived. And what we've found is those brain cells regrow their axons. They regrow their axons all the way to their appropriate target and appear to make a difference. They make the behavior of the animals better. So this was something that was totally unanticipated, and from our point of view, it offers a very substantial new promise in the treatment of Parkinson's disease. Most of the focus has been on trying to replace the brain cells that have been lost, and so that's why there's been so much, and it's very important, talk about transplantation. In years past, fetal tissue cells were used as transplantation. That's not done a great deal in the States anymore. Now most of the focus has been on stem cells, and the effort is to try to replace the brain cells that have been lost. But the approach that we're developing utilizes the patient's own brain cells that are already there. They're still there. They're surviving, but they've lost their axons, and now induces them to regrow those axons and function normally once again. It's become clear that at the time patients are diagnosed with Parkinson's disease, it's only 30% of the brain cells that have been lost. 70% are still there. And that's part of my point about if we can get axons to regrow, there are many neurons that are still there waiting, hopefully, to be rejuvenated. So for many, many years in the textbooks, even now, you pick up a textbook and you read about Parkinson's disease, and it will say that by the time these patients present with their disease, 80% of the dopaminergic system has been destroyed. It's degenerated. And it is simply not true. There are two components of this system. One is the cell bodies, and the second are the axons. And the axons do take the brunt of the injury. So when these patients are first diagnosed, and this has been shown very clearly by PET scanning studies and by post-mortem studies using very sophisticated techniques, that as far as the axons are concerned, at the time these patients are diagnosed, it's maybe about 50% are lost. So it is substantial, but there's a very significant window for preservation of what remains, and we believe possibly for rejuvenating the axonal system. Now, at the level of the neurons, it's only about a 30% loss. 70% of those neurons are still there. So based on more modern techniques, the estimate of the number of neurons lost is only 30%. So we're looking at a far more hopeful situation than many patients have been led to believe is the case. The idea that there's an 80% loss is an antiquated idea. It's not correct.
Perhaps the most dramatic and almost science fiction-like breakthrough recently is something called gene repair. We now have a technology, it's called zinc finger technology. Don't worry about the term. But basically, they're like molecular scissors. And you can design these molecular scissors to go in, and we have, by the way, 3 billion base pairs in our whole genome. And we can actually train these scissors to go in and find one base pair and snip it out and repair a gene. And we can even go further. We can not only remove a gene that's not working, we can replace it with the healthy gene. And if you'd told me that would be physically possible 5 years ago, I absolutely wouldn't have believed it, and we can do it now. The first implication is that when you're doing science, if you have 2 cell lines and you get a different result in your experiment from one from the other, well, if those cell lines came from different people, you don't know, was it the different people or was it your experiment? We can actually now take the same cell, put the mutation or variation in, or take it out. These are called isogenic cell lines, but all that means is these cells are identical twins except that one little change. So anything you see with that change is real. So that changes science in terms of having the perfect control. Someday it could lead to actual correcting genes, humans with genetic diseases actually going in and correcting those genes, which is something, as I said, I did not see in the future, at least not in my lifetime, and we're right on the top of it now. In fact, there's a whole new technology that may replace one I was just talking about that's even better. We found that one aspect of Parkinson's which was little known up until 10 years ago, is loss of innervation of the heart. Turns out that when you lose your innervation of your heart, which is almost universal in Parkinson's, your heart rate variability changes. Your heart is varying second by second, and we can measure that 14 different ways. And it turns out we can measure that with a simple EKG. Most EKGs are digital. All you would have to do is run an EKG 5 minutes instead of 1, calculate HRV. Patients that have decreased HRV could then go on for a second level of tests, eventually scanning and early diagnosis. And if we can get this into a general practitioner's office, we can do that. That's called secondary prevention. If you can get in there, yes, the disease has started, but it's not causing symptoms, you could actually prevent the symptoms of the disease. So this is a very exciting new direction that we hope to be able to get into the primary care physician's office so everybody starts getting screened in, say, their 50s and on. Another test that may be very useful early in Parkinson's is loss of sense of smell. Oddly, Parkinson's begins in the olfactory bulb, and we can measure that with a real simple scratch and sniff test. And that's another thing that we would like to get into a family physician's office. In fact, that takes no time. All he has to do is hand the test to the patient. They can take it home, do it on their own, and mail it in. So it's almost no extra burden on the primary care physician. So if smell's down, if heart rate variability is down, we're starting to build a real case. You may have early preclinical Parkinson's disease.
These patients have motor symptoms from Parkinson's disease, but also non-motor symptoms. Now, the motor symptoms include the features that everyone knows the best: the tremor, the stiffness of the limbs, the slowness of the movement, and the poor balance. They tend to fall easily. So those are the motor problems. The non-motor problems include forgetfulness, depression, problems with sleep, problems with what we call autonomic function, which refers to maintaining your blood pressure, and bowel movements. These are autonomic functions.
We've got 5 drugs that are currently being tested to slow or halt the disease. There are many more in the laboratory. If we get a hit and we can screen the general population for early or premotor when there's little or no damage, we could really, I think, do secondary prevention. That's the future I see.